Hello again. Now we're on the final part of Chapter 2, Part C, Chemistry Comes Alive. Organic compounds. Organic compounds contain carbon. So all these carbon-containing molecules that we're going to be talking about are organic compounds. Carbon dioxide and carbon monoxide, even though they contain carbon, are not considered organic. But remember that the carbon atom is electroneutral. That means that in order to fill its valence shell, it will form 4 covalent bonds. Excuse me. It will share pairs of electrons with other atoms, sometimes with single bonds, sometimes with double bonds. But always it's going to have 4 covalent bonds. Organic compounds are unique to living systems, and the organic compounds that we're going to be talking about include carbohydrates, lipids, proteins, and nucleic acids. Sometimes these are referred to as the molecules of life, and they're often quite large molecules. Organic compounds are usually polymers. They're big. So a polymer is built with several small monomers that are joined together. I think of a polymer somewhat like a train. So a train is built with smaller subunits, train cars, and so the train car would be the monomer. And each train car is separate, but when they're joined together, it makes the entire train. The way monomers are joined together is through that process called dehydration synthesis, which I mentioned in the last PowerPoint. But we're going to look at that, dehydration synthesis, and then they're broken apart through hydrolysis. And figure 2.14 illustrates these processes. Dehydration synthesis, so if you think of dehydration, it's taking out a water molecule. So with 2 separate monomers, when you take an oxygen atom and a hydrogen atom off of this one, and then you take a hydrogen atom off of this monomer, and that is clipped out using an enzyme, this water molecule is released, and now these 2 monomers are going to be linked by a covalent bond. Hydrolysis is the opposite. So if you think of hydro, that's referring to water. Lysis means breaking apart. So this is when monomers are released by the addition of a water molecule, and it adds the OH to one monomer and the hydrogen to the other. So here's 2 monomers linked by a covalent bond using a molecule of water. They will be chopped apart. Oxygen and hydrogen atom put onto this one, and a hydrogen atom put onto that one. This happens during digestion. Within the gastrointestinal tract, you'll see that there are a lot of enzymes when we get to that unit, which is not until Human Anatomy and Physiology 2. But there are a lot of enzymes that are designed to chop our food molecules, large complex molecules, into their subunits or monomers. Here's an example, dehydration synthesis of sucrose and then its breakdown by hydrolysis. Sucrose is table sugar. It's a disaccharide, meaning 2 smaller sugars are put together, 2 simple sugars, and then it's showing you right here the 2 simple sugars are glucose and fructose. And here's showing when a water molecule is released. Here's an oxygen-hydrogen atom over here. And then there's a hydrogen atom over here on the fructose, and that is cleaved off, and that allows those 2 simple sugars, glucose and fructose, to be joined together to form sucrose. This happens quite often in plants when they're making their sugars. Now, just on going the other way, excuse me, and we have an enzyme actually called sucrase that when we when we have sucrose in our diet, that enzyme will actually cleave apart these 2 simple sugars using hydrolysis. Think of hydrolysis. So using a water molecule, and obviously this is happening in water. All of our body cells, in and around our body cells, is plenty of water. So a water molecule is used to chop sucrose into glucose and fructose. So we have an oxygen and a hydrogen added onto this side is going to make the glucose, and then a hydrogen atom added to here, which makes the fructose. So now the sucrose is chopped into these two simple sugars. Simple sugars can actually be absorbed into the body, into our body cells. And you may remember cellular respiration, where glucose is used for cellular energy. So of these four groups, the first major group is called carbohydrates, and sometimes they abbreviate that CHO for carbohydrate. This includes starches, sugars, and starches, and the atoms involved are carbon, hydrogen, and oxygen. Carbohydrates include the monosaccharides, or simple sugars. Saccha means sweet, so that's why you may have heard of that artificial sweetener saccharin. But monosaccharides would be a simple sugar. Disaccharides, remember this prefix di means two, so disaccharides are double sugars. Poly means many, so when many simple sugars or monosaccharides are joined together, it forms a large complex carbohydrate, and we call that a polysaccharide. So what are carbohydrates for? The functions of carbohydrates is that carbohydrates provide our major cellular fuel, particularly the simple sugar or monosaccharide glucose. Some in nature are structural molecules. Actually, animals have a complex carbohydrate known as glycogen, but that's a short-term energy storage. Plants and insects actually have some complex carbohydrate that forms their structure, and we will see those coming up very soon. But let's start with monosaccharides. Monosaccharides are simple sugars. They consist of 3 to 7 carbon atoms. This is the monomers, or the simple sugars are the monomer of the complex carbohydrate when they're joined together. These monosaccharides include 5 carbon sugars. They're called pentose sugars, and it's got a carbon chain where the carbons are connected together, but they actually form a 5-pointed structure. Looks kind of like a house. You'll see that in a minute. The 5 carbon sugars include ribose and deoxyribose, and you may recognize these because ribose is the sugar that forms, along with other subunits, ribonucleic acid, RNA, and deoxyribose is the sugar that is in that big complex molecule, deoxyribonucleic acid, or DNA. Hexose sugars have 6 carbons, and these also go into a ring structure. Glucose is the one that our cells often prefer to use for energy, and when we say you have your blood sugar, a blood test to measure your blood sugar level, that's referring to glucose. And here is a figure that shows some of these monosaccharides or simple sugars, the monomer of carbohydrates. These are hexose sugars. Hexose means 6. So we've got glucose, fructose, and galactose. I'm not going to ask you to memorize the chemical structures here, but these are called isomers. They are all the same atoms, but they're arranged somewhat differently from one another. And then here is a figure showing you the pentose sugars, deoxyribose and ribose. And surprisingly enough, deoxyribose has 1 less oxygen atom than ribose. So the names actually somewhat make sense. Disaccharides are double sugars. Double sugars are too large to pass through cell membranes, and important disaccharides are sucrose, maltose, and lactose. And again, you should remember these examples. You're not going to need to draw the chemical formulas, but here's a figure that shows you sucrose. Sucrose, table sugar, is formed when glucose and fructose join together. 2 glucose molecules joined together forms maltose, and lactose is composed of galactose and glucose joined together. Lactose is milk sugar. Polysaccharides. These are large polymers of monosaccharides. So many monosaccharides joined together will build a polymer, a polysaccharide. Examples are starch in plants. Starch is actually a structural polysaccharide. Beep! Polysaccharides are polymers of monosaccharides. Examples include starch in plants. Starches are actually the energy storage for plants. So a potato plant, for example, stores energy in the form of a potato. Potatoes have a lot of starch in them, and so to meet the growth needs of that plant can actually have some enzymes start breaking down the starch into sugars, which can be used during aerobic cellular respiration. Cellulose forms the plant cell walls. So cellulose forms is a structural polysaccharide, and it forms things that we think of such as stems, and also tree trunks are cellulose, and we make cardboard and paper out of that. Glycogen is a polysaccharide that is a short-term energy storage in animals, and chitin is a structural polysaccharide that forms the exoskeletons in insects and crustaceans like lobsters and crabs, etc. Polysaccharides are very large and they're not very soluble. And this is a figure showing you glycogen. It's a polysaccharide that's formed from many glucose units that are joined together. Lipids. Lipids, or fats, contain carbon, hydrogen, oxygen, and sometimes phosphorus. These are insoluble in water. Remember, fats and water do not mix. The types of lipids include triglycerides, or neutral fats. This includes animal fats and oils, vegetable oils. Phospholipids is another type of lipid, steroids, and eicosanoids. Triglycerides, or neutral fats, are called fats typically when they're solid and oils when they're liquid. They're composed of 3 fatty acids that are bonded to a glycerol molecule. So that's why these are called triglycerides. The main function of triglycerides is energy storage, that is long-term energy storage in animals. The triglycerides form our adipose tissue, which is a long-term energy storage molecule. It also provides insulation and protection. So with insulation, sometimes if someone is really, really thin, they don't have much body fat, they will often become cold much more quickly than someone who has a layer of adipose tissue. Also, in a hot environment, they will become overheated much more quickly. So fat is your friend. Also, it forms protection. So the adipose tissue that is in our body, we don't see the pockets of fat that are inside our body typically, but there's fat pads around your eyeballs in their orbits that protects them from trauma. There's also fat pads around our kidneys, which protects them as well, and in many other places. And this is a picture of a triglyceride and how it's formed. Here's the glycerol molecule right here, and the 3 fatty acid chains that are joined to it through this dehydration synthesis. So taking out a molecule of water, a hydrogen from here, hydrogen and oxygen atom over here, it will join each of these fatty acid chains to the glycerol molecule. So this can actually, this is actually what's going to form the neutral fats, or adipose tissue, animal fat, and also vegetable oils. There is a little difference between some of these fatty acid chains that you see. A saturated fatty acid has a single covalent bond between the carbon atoms. So therefore, they have the maximum number of hydrogen atoms attached to them. And you can see over here, remember, each carbon atom will make 4 covalent bonds. And so we see this first carbon atom here has an oxygen atom bonded to it with a double bond, and then it has a bond over here where it's joined to another part of the molecule, and then over here to another carbon atom. So typically, a carbon atom may have a hydrogen atom joined this way and a hydrogen atom drawn down there. They just draw it as CH2, and then there's a carbon atom joined to the next carbon atom. Sometimes we call it a carbon backbone because when carbon atoms are joined to one another, it's somewhat is similar to how our vertebrae are joined to one another in our backbone. But if you look along here, this fatty acid chain is straight. It's saturated with hydrogen atoms. This particular fatty acid is actually we can call it an unsaturated fatty acid because look, there is one double bond here between this carbon atom and that carbon atom. So therefore, there's no more bonds left to join to another hydrogen atom here. So that would be unsaturated. And so when we say polyunsaturated, that would be many of these double bonds here. But an unsaturated fatty acid is when one or more double bonds occurs between carbon atoms, decreasing the number of hydrogen atoms. And so typically, the unsaturated fatty acid will be liquid at room temperature. An example, of course, would be the vegetable oils like safflower oil or canola oil. It's interesting what we hear about a lot, trans fats, which are bad for us, because when we cook with them, it can yield some harmful byproducts. But a trans fat is when scientists they'll take like an oil and they will process it and force more hydrogen atoms to be joined onto these fatty acid chains. So it will turn an oil into a solid. And that was the great discovery of margarine. I think it may have been back in the 1950s, because they were kind of wondering if eating a lot of butter was causing people to have more arterial sclerosis, because the saturated fats, now they're beginning to have a different opinion about it, but at that time it was like by having a lot of saturated fats, it led to an increased risk for arteriosclerosis, which could lead to strokes and myocardial infarctions, which is a blockage to one of the coronary arteries that would lead to what we call a heart attack, etc. So anyway, they created margarine, and it was artificially hydrogenated so that at room temperature it stayed solid. But then later on, people were discovering that when you cook with margarine or even having these trans fats in your diet, it's not so healthy. So now you might buy a product and it will say no trans fats in it, and so that's a good thing. Omega-3 oils, these are commonly found in fish, and these are considered healthier than the like other animal fats, the triglycerides from a source such as in hamburgers or chicken. Okay, the second type of lipid would be a phospholipid, and you've heard about phospholipids because this forms the cell membrane, the plasma membrane around the cell, and then the membranes even within the cells that form the cellular organelles, such as the nucleus, endoplasmic reticulum, etc. Phospholipids are modified triglycerides. So there's a phosphate head, which is polar. That means it's compatible with water. Keep in mind, these little water molecules themselves are polar, and so a polar molecule would be water compatible. Now, the fatty tail is not H2O compatible. This is remember, oil and water don't mix, so the fatty tail regions are not water compatible. This phospholipid membrane is actually a bilayer. And this is the figure of a typical phospholipid. So here, this is the phosphorus-containing polar head, and it's showing it right here. And then here is just the glycerol backbone, but over here you have your fatty acid chains, and these are non-polar, so they do not mix with water. And keep in mind the cell membrane it's actually two layers. Toward the outside of the cell, you have your polar heads that are lined up along the outside surface of the cell membrane, and then the fatty acid tails go toward the center of that membrane, and then you have another layer where your polar heads are oriented toward the inside of the cell with the nonpolar tails pointing toward the inside of that membrane. And we'll come back to cell membranes later. But steroids are the third group of lipids, and these are interlocking four-ring structures. And this is steroids are based upon the molecule cholesterol. Vitamin D and steroid hormones are all necessary molecules for our body. Cholesterol has actually gotten a bad name, but it's absolutely essential that we have some cholesterol so that we can make our steroid hormones. When we get to Bio-264, or ANP2, we have a whole unit on the endocrine system, and you will see that there's a category of steroid hormones that includes the hormones that are produced by the testes and by the ovaries, and also the adrenal cortex that produce very powerful hormones that are based on cholesterol, and they're called the steroid hormones. Also, bile salts have some cholesterol in there. Keep in mind the steroids have the cholesterol is the most important. Sometimes these are called sterols as well. And here's a picture showing you a steroid. And this is the cholesterol as the basis for all the steroids formed in the body. I'm not going to ask you to memorize the chemical structure. Just keep in mind this is a type of a lipid. And then finally, the fourth major type of lipid would be the eicosanoids. There are many of them. Just keep in mind this word is one of the types of lipids, and the major one is called the prostaglandins. And we'll come back to talking about prostaglandins later, particularly when we get to the reproductive system. But we talk about, later we'll talk about blood clotting, control of blood pressure, inflammation, and uterine contractions. But keep in mind, prostaglandins are actually active in all of these processes: blood clotting, control of blood pressure, inflammation, and actually with uterine contractions. That's where I actually bring up prostaglandins later. Other lipids would be lipoproteins. These transport fats in your bloodstream, and also your fat-soluble vitamins, which are essential for your health: vitamins A, D, vitamin E, and vitamin K. Now we're on proteins. Proteins are, there's so many proteins. They're extremely varied. The human body is made up of somewhere between 17 to 18,000 different kinds of proteins. I am not going to click on this link because I'm not sure if I'll be able to get back, but you can look at this link on your own, and it's actually, if you've heard of the Human Genome Project, which is studying the genetic makeup and analyzing our DNA, there's actually the Human Proteome Project as well that is studying all the variety of proteins in our body, which includes enzymes, structural proteins, etc. So keep in mind, proteins are quite complex, they're large, and we have many, many kinds of proteins that makes up our phenotype. What we look like, how our body functions, is all based on our collection of proteins. And proteins are built with amino acids. So the protein would be a polymer, a big complex molecule, and it's made with little monomers or subunits called amino acids. There's 20 different kinds of amino acids in nature. I'm not going to have you memorize all of them, but keep in mind that every amino acid has an amine group and an acid group, and it kind of makes sense because it's called an amino acid. The amine group has a nitrogen atom in it. And so when our body is breaking down amino proteins and then breaking down those amino acids, we actually need to get rid of this nitrogenous waste. The breakdown products of these can be toxic to our body. So we have specialized systems in our body to deal with that. But if you'll take a look, here's a nitrogen atom. This side of an amino acid is called the amine group, and this side is actually is an acid. It's a carboxyl group. That's what this is called, and it is acidic. So this is showing you glycine, which is the simplest amino acid, with the amine group and the acid group, as all amino acids have. This one, it's remainder. See the R, that means the remainder of the molecule. It's only a hydrogen atom. And then we get to more and more complex amino acids. Here's aspartic acid. There's its remainder. Lysine, which is quite a bit bigger. Cysteine, which actually has a sulfur atom in it. But every amino acid has the amine group and the acid group. And actually, when we're joining these amino acids together in order to form a polypeptide, polypeptide is referring to the type of bond that occurs. These polypeptides will fold up into complex structures that we call proteins. So here's the carboxyl group, which will join to the amine group of the next amino acid, etc. And this is actually illustrating that. So on the carboxyl group, we've got an oxygen, a hydrogen atom, and then on the amino group right over here, the hydrogen atom, we do dehydration synthesis, where that water molecule is chopped off in order to join these two amino acids together, and this is called a peptide bond right here. Hydrolysis is actually when a molecule of water is used in order to chop these two amino acids apart into their amino acids. So there is that illustration. Remember, proteins are quite complex, and they have different levels of structure. The first structure, or primary structure, would be the sequence of amino acids that forms a polypeptide chain. So it's which amino acids are used in order to make this polypeptide chain. A secondary structure would be when that chain of amino acid will, what shape it assumes. It will either be a spiral, it's called an alpha helix. Alpha helices are actually, helices is the plural form of helix, or a beta sheet, which is kind of weird. There's only two major shapes. It'll be either this helix shape or it will be a folded sheet. So here's the alpha helix. The primary chain is coiled to form a spiral structure, which is stabilized by hydrogen bonding between various regions on this polypeptide, on the amino acids actually. The beta sheet is when the primary chain zigzags back and forth to become a pleated sheet, and the adjacent strands, of course, are held together by hydrogen bonding. Tertiary or third level structure. This is going to be superimposed on secondary structure, and it's when the alpha helix or a beta sheet is folded up to form a complex globular molecule that's going to be held together by intramolecular bonding. This is showing you an example of a tertiary structure of prealbumin. Beep! Figure 2.19c shows you a level, the tertiary level. Beep! Tertiary structure. This is actually the third level of protein structure, and you can see this is superimposed on a secondary structure, either an alpha helix or a beta sheet, which is going to be folded up to form a compact globular molecule. I think of this structure as, say you take a helix, like a slinky, and it's going in a helical spiral shape. So that spiral shape would be the secondary structure, and then if that slinky gets twisted up upon itself, that would be considered the tertiary structure, or third level. And this is showing you an example right here. The quaternary structure means the fourth level. This is when two or more polypeptide chains, each with its own tertiary structure, combines to form a functional protein. So let's just say we had the slinky example. You have two slinkies, and they are in their typical spiral or helical shape. Each one gets folded up on itself, and then they get twisted together. So this particular example is showing a quaternary structure of the prealbumin molecule. And so there's many proteins that are quite large, something like hemoglobin. It actually has 4 polypeptide chains that are folded up in their particular structure, and they all come together to form a large protein. Quaternary structure would be with the 2 or more polypeptide chains that join together. Table 5.1 is an overview of protein functions, and as you can see, there are many types of proteins with various functions. There's many kinds of enzymatic proteins that will accelerate chemical reactions. Remember, they catalyze chemical reactions, allowing them to happen in a reasonable amount of time. Example, some of our digestive enzymes, actually all of them are proteins, are structural proteins. Silk fibers, that would be from the silkworm. Collagen and elastin forms our connective tissues. Keratin in our hair, in our horns and feathers, and other skin appendages such as nails. Storage proteins, here's your examples here. Transport proteins, things like hemoglobin or other proteins that transport substances in our bloodstream. Hormonal proteins. Now, there are some of our most powerful hormones, and actually all of them are powerful. I mentioned before steroid hormones. There are other hormones that are based on proteins, things like adrenaline or insulin. These are actually hormones that are proteins. So that's showing you insulin right here. Receptor proteins. These are proteins on our cell membranes that will respond to stimuli. Contractile proteins like actin and myosin in our muscles. We're going to be talking about that quite a bit when we get to skeletal muscle functioning. Defensive proteins are antibodies, are protein molecules. So there are many of them. There's a different form of protein. There's 2 major types of shapes of proteins. There's fibrous proteins and globular proteins. Fibrous proteins are long, kind of like strand-like proteins, and these are typically our structural proteins. They are strand-like, they're water insoluble, and very stable. They actually achieve mechanical support, and they provide strength, tensile strength. Examples would be keratin, which is in our hair, elastin and collagen, which is these are proteins that are in our connective tissue. Our bones, for example, have structural protein collagen, and in and among those collagen fibers are calcium phosphate crystals that make bone hard. Globular proteins, I always think of a little glob shape. These actually have they look like spherical proteins. They're very compact, but they actually have a very specific shape to them. So they are water soluble, and they're sensitive to changes in pH and temperature. So specific functional regions on these globular proteins are called active sites, and that's really important for the functioning of these particular proteins. Examples are antibodies, hormones, and enzymes. Protein denaturation. Now, protein denaturation just means that protein has been changed in shape. When it comes to our globular proteins, if you change the shape of that protein, it will lose its activity because you're losing that functional 3-dimensional shape, and you're destroying the active site, so it can't work anymore. Could be a change in pH or a change in temperature. It's usually reversible, but sometimes it's irreversible if it's extreme. Now, other structural, any kind of protein can be changed in shape through heat or change in pH. But when it comes to our globular proteins, those are the proteins that will actually lose their function because it's their shape that's so important that allows them to function properly. And this is just showing you cottage cheese. So this milk protein casein is actually denatured at a pH of 4.6. So it's an acid that's subjected to this milk protein, and it creates curds and whey. Whey is the liquid part, and these are the curds. Over here, this is showing an old-fashioned way that hairdressers used to use to get a permanent curl to the hair. And I think they may have had some accidents, which is why they stopped using the heat. But they would use heat, and once they subjected the hair to the heat, then you take the curlers out, that hair would be permanently curled. Now, obviously, and thankfully, your hair grows back. If it's damaged or curled to the point where you don't really like it, eventually your hair will grow out. But we actually use some heat if you want to use a gentle heat, like with a curling iron or something, to change the shape of your hair. And it's really affecting the shape of those proteins, keratin primarily. Enzymes. Enzymes are globular proteins that act as biological catalysts. They regulate and increase the speed of chemical reactions. And what they actually do is they lower the activation energy and increase the speed of a reaction. And literally, this could be millions of reactions per second. Reactions per minute. So lowering the activation energy, it just allows that chemical reaction to happen much more quickly. And when I was first studying this activation energy, the enzyme actually lowers the amount of energy needed to start that chemical reaction. I actually think of enzymes as like little workers at an assembly line in a plant that's manufacturing something. So the enzyme will actually facilitate a certain chemical reaction. Without the enzyme, you have a lot of energy that's required to get that chemical reaction to go. This one, not so much, because the enzyme is present, and so it's just much easier to allow that chemical reaction to take place. Okay, enzymes, what are they? Some of them are holo enzymes or hollow enzymes, and that means they have two parts. There's a protein portion, and then there's a cofactor, often a metal ion or some type of a coenzyme, which is based on a vitamin. So if you were wondering why you need vitamins, vitamins are often associated with an enzyme, so a certain kind of chemical reaction facilitated by a specific enzyme. So enzymes act on a specific substrate. If you'll remember, a substrate is the raw material on which the enzyme acts. Enzymes usually end in ASE, not always. However, when you see something that has an ASE at the end, it's most likely an enzyme. Some enzymes have different names, but we often see ASE at the end, and the beginning part of the word will be named for the reaction that's catalyzed. This is showing the mechanism of enzyme action, where you have, let's see if there's there's the step art. So the substrate, here's the enzyme, the purple thing. The enzymes are going to bind at the active site, temporarily forming an enzyme substrate complex. Then the energy is absorbed in order to create a bond here. The enzyme substrate complex undergoes internal rearrangements that form the product. And then in this example, they're showing building putting together two amino acids to eventually form a polypeptide. But this is showing that once these are joined together, the enzyme releases that product. And it will be passed to the next enzyme, which will then work on that particular product, and eventually you'll have a final product. Now we are on nucleic acids. DNA, deoxyribonucleic acid, ribonucleic acid, RNA, and ATP are the three nucleic acids that you need to learn about, which you already know. This is somewhat review. DNA, RNA, and ATP. Particularly the first two are large polymers. DNA and RNA are quite large. ATP is also somewhat large. Remember the name for this one? Adenosine triphosphate. So all of these are built with a subunit that we call a nucleotide. And here is showing you the structure of DNA, and they're showing you the nucleotides. DNA has adenine, thymine, cytosine, and guanine. And we often just show the letters here. A for adenine, T, thymine, cytosine is C, guanine, G. And DNA is a double helix. So it has two strands, okay, where you've got your sugar phosphate side chain. There's a sugar. Remember, in DNA it's deoxyribose. There's the pentyl sugar. You've got a phosphate group over here, and then you have your nitrogen. We call it nitrogen-containing base because it contains nitrogen. So this is adenine. And then this side chain has the sugar phosphate side chain, and coming out toward the center, we've got thymine. And it was discovered over many years and with a lot of hard work by various scientists. We do have this rule where adenine pairs with thymine and vice versa, and cytosine always pairs with guanine and vice versa. And scientists are now analyzing all of our chromosomes, and they have come up with the order in many enzymes, actually the order of our whole genome. And we call these actually base pairs because even though these are nucleotides, because they have that nitrogen-containing base that makes them different, we could call them bases for short. So here's showing the sequence of DNA. Ribonucleic acid, or RNA, it also has 4 nitrogen-containing bases: adenine, guanine, cytosine, but instead of thymine, it's uracil. So it's slightly different in that of the 4, it's not thymine, but uracil. The pentyl sugar on the side chain is ribose, not deoxyribose. And this is a single-stranded molecule, and it's mostly active outside the nucleus. RNA is actually formed off of the DNA template, and then it will exit the nucleus and then go out to the ribosome to direct protein synthesis. There's 3 types of RNA: messenger RNA, little m RNA, transfer RNA, little t RNA, and a ribosomal RNA, or lowercase r RNA. ATP is actually adenosine triphosphate. The structure is, it has adenosine, which is formed from adenine, and remember that's a nucleotide, that's why ATP is in the nucleic acid family. This is the molecule that with which, or in which, chemical energy is captured. When glucose is chopped down, ATP molecules are produced, and ATP will then be used in areas of the cells where energy is needed. It directly powers chemical reactions. So the energy from sunlight or from food will actually, and obviously for sunlight this would be in plants, ADP plus inorganic phosphate can be joined together to make ATP. ATP is will now be energy that's available for cellular work and chemical synthesis. So all the work that cells have to do, anything that requires energy, will be provided with energy with using this molecule ATP. And here's the structure of ATP. Here's adenine, and then you've got a ribose sugar right here. This is adenosine, and here are your 3 phosphate groups. That's why it's called triphosphate. This very last bond is extremely high energy bond, and then when it's hydrolyzed or chopped off, this terminal phosphate group, that means the last one, it will then release energy that can be used to do cellular work. So what's the function of ATP? It actually phosphorylates other molecules. The terminal phosphates can be transferred to and energize other molecules, and breaking that phosphate bond will be actually that's the process that can be used to drive cellular work. And here are some examples. Transport work, this is when things can be actively transported across the cell membrane. You can see mechanical work, and this would be for muscle contraction, and even with chemical work, when we are having energy absorbing chemical reactions in the body. In order to do that chemical reaction, it may need some energy, and that will be provided by ATP. And that's the end of chapter 2. Have a wonderful day. I will see you later.